Thank you all, and thank you, Christian, for inviting me. And it's great to be back. I was here 7 or 8 years ago or something. So yeah, this is like, and now for something completely different. But this crowd of all crowds will totally get it. So who are the biggest troublemakers in evolutionary biology? And I think we can all agree on number one. Probably good agreement on number two. And here I declare a tie. And also, but chemical engineers. So this is my colleague and collaborator and buddy Tenmei Lele, who formerly at the University of Florida, and now he's an Aggie, Texas Aggie, Texas A&M. And this project had its origin when the UF Medical School was shooting for like one of these big NIH Centers of Excellence grants. So they found everybody who had a grant that sounded anything like cancer. And I had a grant on mutation in worms. So they rounded me up and said, sure, yeah, I'll give my 3-minute spiel. And I gave my 3-minute spiel on mutation accumulation in worms. And afterwards, Tenmei came up and said, oh, Charlie, that was so cool. He said, hey, let me run something by you. If I was to put cell culture on soft substrate and stiff substrate, growing, animal cells on soft substrate and stiff substrate, do you think they'd evolve? And I said confidently, yes. And that was the origin of this project. So anyway, so I've got two stories here. I suspect the one story will suck up 98% of the time. This was published already back in 2021 in MBE with Tenmei and Prabodja Perkias, that was the lead person on the project, and then my at the time graduate student and now old buddy Ayush Saxena. So anyway, the idea is that so Tenmei coined the term mechanoevolution. So the idea is that the mechanical properties of the environment are a ubiquitous agent of natural selection. And if you don't think this is true, walk out on a patch of quicksand, right? So here, like the tadpole and the adult frog experience a very different mechanical environment. Jellyfish experience a very different mechanical environment under certain circumstances. E. coli, if you grow E. coli in liquid culture and you plate it out on an agar plate, the cells themselves will have very different properties. Because there's some, this is Tenmei's slide, and I don't know what this stuff is, but don't worry about it. So, this is just kind of a summary of the various mechanical forces that act on a cell. So if you can smush the cell, it's got layers of tissue below and beneath, compression, gravity, shear, surface tension, all these various kinds of mechanical forces act on the cell. And presumably they impose natural selection of various types. So, here are some examples of animal cells in vivo. So, mammary gland development. So the ECM is the extracellular matrix, and I'll use that term repeatedly. So there's a stiffness gradient in the extracellular matrix of the developing mammary gland, and how the gland depends on this gradient in order to develop properly. Angiogenesis, blood cell development, same thing. There's this stiffness gradient in the extracellular matrix. Limb development in mouse, there is a mesodermal stiffness gradient. Mesenchymal stem cell differentiation, and for our purpose, for our long-term purposes, the one we're most interested in is in tumors. So a tumor himself gets stiffer as it grows, right? And the reason you can tell, hey, I got a lump in my breast or my head or whatever, right, is because you got this hard lump because the tissue is stiffer than the surrounding tissue. And also, if you think about metastasis, if you have a cell, for example, suppose you have a breast cancer cell and it metastasizes to the bone. Well, the bone is very much stiffer than even the surrounding breast. So the cell will have a very different mechanical milieu. Or if a tumor cell migrates to something soft like the brain, so the idea being it might experience either a much stiffer or much softer environment than the environment it is locally adapted to. So this is the premise of Tenmay's idea. So here are some more specific examples that are directly relevant to the project I'm going to tell you about. So if you culture, grow cells in culture on substrates of different stiffnesses, cell area. So you say, okay, we just take a picture of the cell and how much area is it taken up. So cell area increases with substrate stiffness up to a point. And again, in most, not all, most cell types. And the cell volume itself tends to decrease with substrate stiffness. And all of these are sort of, usually, but not always kind of things. Cell mechanics, attraction stresses, cells, don't just sit there like a lump. They can move around, and they think about a developing organism, those cells are moving. And so the traction they get, they get better traction on a stiff substrate than on soft substrate. And the substrate stiffness itself can affect the cell stiffness. So if you poke a cell, how much will it displace will depend, sometimes at least depend on the substrate. Cell motility, like I said, cells will move, and some cell types or many cell types exhibit durotaxis. So depending on what the the cell's idea of an ideal substrate stiffness is, they will move toward or away along a gradient of stiffnesses. And here, for the purposes of evolutionary biology, cells typically divide faster, they grow faster on stiff substrates. Again, not but up to a point, right? And often they cell, in a cell, for context of cell differentiation, cells differentiate best on substrates, of the appropriate stiffness. Okay, so this is really kind of an invisible pointer here, I think, and also it's not very reactive, but now it's stuck. Maybe if okay, so this is kind of a weird thing. So cells have what the people in this line of work refer to as mechanical memory. So for example, primary fibroblasts, and this experiment I'm talking about, these are not primary fibroblasts. These is an immortalized cell line. But primary fibroblasts, if you culture them on the stiff substrate, and then you move them onto a soft substrate, they will retain that suite of stiff substrate phenotypes for 4 or 5 or so cell divisions before they revert to their canonical soft substrate phenotype, and vice versa. And that's kind of weird. And there's a fair bit is known about the regulation of that. It's very small RNAs, which are any worm biologist would predict. Everything is due to small RNAs. But okay, so here I don't have to explain to this crowd when I'm talking to chemical engineers. I have to patiently explain the idea of phenotypic plasticity. You guys know what phenotypic plasticity is. So, and it's usually applied in the case of autonomous organisms, right? If I'm a Daphnia and I smell the scent of Chaeoborus larvae in the water, I grow one of these little helmets, right, in the next generation. And I say, but the principle applies equally to somatic cells. Cells will take on, those examples I just gave you, those are examples of phenotypic plasticity. Cells with the same genotype will develop different features depending on the environmental context. It is usually assumed, at least in the circles I travel, it's usually assumed that plasticity reflects evolutionary adaptation. But this need not be true. Okay. So and this is sort of recapping what I told you a few slides back, and just sort of I've got a fibroblast, and I say, okay, if I grow it on stiff substrate, it will grow fast. It will it will spread a lot. I will have a lot of these highly spread, fast growing cells. YAP locally, so yes associated protein that is associated with cell signaling. So YAP is located in the nucleus on a stiff substrate. And if you grow the same cell on a substrate, the YAP stays in the cytoplasm.
What does it mean?
That's a good question. It is so YAP is, I mean, it's associated with cell division and cell growth. And the honest answer is I don't really know what it means. I'm not a cell biologist. It is associated with, well, you'll see some results and you can judge for yourself what it means. Actin fibers, if you grow a cell on stiff substrate, it has, more actin fibers than it, or more or no actin fibers, et cetera. And there's this whole syndrome of phenotypic traits leading to faster growth rate on stiff substrate than on soft substrate. Okay, so, and the first argument here, I don't really believe. So if you think about it, so if you have a plastic phenotype, the Daphnia grows a helmet when there's Chaoborus larvae in the water, and it doesn't if there's not. So one possibility is all these different morphologies, these different plastic morphologies. These are optimal. This is the optimum phenotype. This is, natural selection wants the cell to do these things. Millions of generations of evolution have resulted in a cell that manifests the optimal phenotype in each environment. And I wrote those words, and I confess, I have a hard time believing it. But if you think about it, there's an awful lot of diversity of animal cell types. Like a squid axon is like 30 meters long or something, tremendous, diversity of animal cell types. So within some parameters of gas transport and everything, I don't think, I wrote that, I say, it sounds far-fetched, but on the other hand, I am unwilling to summarily dismiss that possibility. The second possibility, which is the one that seems much more likely to me, is that these, the suite of plastic phenotypes represent trade-off. This is the best, the multivariate optimum, if you will. This is the best the cell can do under these contexts. And for example, if you think about a cell growing on, a stiffness gradient, right, improving performance on as the substrate gets stiffer. If there's a trade-off, you may not necessarily be able to do so well on the soft if you go back, right? So, and this implies kind of a directionality, at least the way I set the argument up. So those are two extreme alternatives, maybe, or it's all random genetic drift. Okay, so here's where you guys are like, all right, now we're talking about something. So what we did is we did the same thing many you guys do, which is experimental evolution. And as you know, you have replicate populations derived from a common ancestor. You propagate them for many generations under well-defined conditions, and the well-defined conditions impose natural selection, right? Which is to say, I said a heritable trait, but a mutation that confers a trait value that is associated with high fitness will increase in frequency, and one that reduces your fitness will decrease in frequency. And normally heritable is equal to genetically variable, although that mechanical memory stuff kind of doesn't, where'd that come from? All right. Okay, so here's what we did. So in NIH 3T3 fibroblast, this is an immortalized cell line that was originally derived from a highly inbred mouse. So originally the cell line was highly homozygous. Diploid, no recombination, right? So this cell culture this line has been maintained for decades on tissue culture plastic. Tissue culture plastic is stiff. So, and for something like 10,000 generations, I am willing to claim that yes, these cells should be close to mutation drift selection balance, right? And these, and these cells show the typical phenotypic plasticity when you culture them on stiff versus soft substrate. Okay. So this, you guys are used to seeing figures like that, right? Oh, it's like I don't know what that was. It said on. But, all right. So Yeah, okay, so we start with an ancestral population of these fibroblasts, and we have two treatments. We have the soft tissue or the soft substrate treatment, that's the red, and we have the stiff substrate treatment, that's the blue. So this is 1 kPa and 308 kPa, kilopascals per whatever. And we start out each of those with 10 replicates per each of which is started with 10,000 cells plus or minus something, right? And then we propagated these cells for 90 cell divisions, 90 generations. And we bottlenecked them. So every 3 days, which is about every 3 cell divisions plus or minus a little, we took 10,000 cells, and then we grew them up to confluence, and I think confluence was about 10 to the 6th. So we had 10 replicate soft selected lines and 6 replicate stiff selected lines, because Tenmei was not especially interested in the stiff selected, and I convinced him to do the stiff selected treatment. Okay, so I mean, we've shown there's this phenotypically plastic response. If the response is deterministic, right? If a cell, no matter what, if you put a cell on stiff substrate and it does this, and if you put it on soft substrate, it does that, the response to the evolutionary response should not depend on whether the ancestral population has any heritable variants in it or not. So we did a control that I suggested we might. So actually, hang on a second, let me back up here. So as a control, second set of replicate lines, but each one of those lines was descended from a single cell. So this, so there is no genetic variation in this population except for new mutations. But and then we grow it up. Now we grow it up to confluence. We do the same thing every 3 days. We pick 10,000 cells. And same treatment, same population sizes, except the controls were started from a single cell, a single genome, as opposed to a genetically variable source population. So okay, so here's result number 1. So again, so the red with my weird pointer here. The red is the soft substrate, and the blue is the stiff substrate. So this initially, the cells grow about twice as fast on the stiff substrate as they do on the soft substrate. Okay, and a week, 2 weeks, 3 weeks. So and then after a week, it's the same thing, and now we see this big jump in growth rate, levels off, and we see after 3 weeks or so there is no further increase in growth rate. And I said, this sure looks like mechanical memory to me. Okay, so the first question we asked, I'll get could a reasonable combination of suppose we are in fact selecting among variants. Suppose there is heritable variants, so could a reasonable combination of rare variants and strong selection produce that response I just showed you? And the answer is yes. So this is the same plot. I'm showing generations on the x-axis and the allele frequency. So this isn't a simulation. This is just a calculation from Haldane's model, formulation of increase in allele frequency under selection. And the black squares there, that's the real data. And the various lines, there are different combinations of initial allele frequency, initial variant frequency, and strength of selection. And the dashed lines, we start from a frequency of 0.1%. The solid lines, we start from a frequency of 1%. What you can see is that those red lines there match the observed data pretty well. So an initially rare variant with a 2-fold selective advantage fits the observed results very nicely. Okay. All right, so here are those the control, the clonal lines. So if you so these, again, there's 10 replicates on the soft and stiff, 6 replicates on the stiff. And so the way I've, yeah, okay, we can see a very, this is the same over 30 days, right? Remember, the variable populations increased rapidly over 30 days, 2 weeks. There's very, again, we start off with that initial fitness difference, and the soft, those are highly non-significant results. So the stiff selected lines seem to be getting a little bit worse, and the soft selected lines seem to be getting a little bit better, but this is nowhere near the kind of result that we saw from the variable population. So what that tells me is the response, the evolutionary response is not due to some sort of deterministic mechanical memory phenotypic plasticity. We are the data, the data do not support that hypothesis. They do support the idea that we are selecting among clones with different properties. And feel free to argue. All right. Okay. So then we say, okay, how fast? So we get soft selected lines. These are in the red. And now we grow, so after 90 days or whatever of selection, say, okay, let's grow these soft selected lines on soft substrate. And now they, as you saw, they grow, they have the ancestral growth rate on the soft substrate. They only grow about half as fast on the stiff substrate. So their growth rate pattern has reversed. The phenotypic plasticity here is reversed, right? And on the answer, they do the same thing on the tissue TCP is tissue culture plastic. Okay, so now this sure looks like the response to this sure looks like the response to natural selection. But somebody might say, well, what about genetic drift? And I would say, in all 10 replicants, are you effing kidding me? But say, okay, if phenotypic plasticity somehow is the cause of this evolution, the only evolutionary force acting on the population is random genetic drift, mutation notwithstanding. So we got a large population. NE is approximately 10 to the 4th. So in 90 generations, we do not expect very much random genetic drift in a population of 10,000. But you say, okay, well, let's not take anything for granted. How much change in allele frequency would be consistent with random genetic drift? So this is where I brought my buddy Ayush into it. So we did whole exome sequencing. In fact, we did the exome sequencing because we were trying to find the genes that underlie adaptation, which I'll have more to say about in a bit. But we had the data, right? So we got two. So what we did, and we didn't have a lot of money to do this experiment, so we're kind of scraping a few bucks together. So we had 10 sets of soft selected lines. So we randomly picked 3 and pooled them, extracted DNA and sequenced. And we randomly took another 3, pooled them, and sequenced. We did pool-seq, which just Vienna is the world headquarters of pool-seq, right? This room is the world headquarters. So that's what we did. And then we did 1 set of 3 for the stiff selected controls because I insisted. And then we sequenced the ancestor, so we would have an idea of what the ancestral genotypes were. So then we simulated this experiment, exactly like probably a dozen of you guys have done in this room, right? So okay, so we simulated the experiment, and there was the experiment, right? And okay, so here's the starting material. So there were about 750,000 segregating sites in the ancestor. And so this is about 1.5% of the sites that we sequenced. And there were about 425,000 of those sites of variants that had minor allele frequency greater than 1%. So, and again, now keeping in mind these are, these are somatic cells. The genomes are completely linked and in complete LD, right? Well, the genomes are completely linked. There is no recombination. So we use short read sequencing, so we can't say anything about haplotypes. So I made the executive decision. We will assume everything's in linkage equilibrium. Because that's the best we could do. So this figure is not the typical figure you would see in populations. So this is an unfolded site frequency spectrum, which this is not the usual way to show an unfolded site frequency spectrum. But we start at, so the allele, okay, I am now pointing over here, but okay, so this allele frequency goes from 0 to 1, right? And the reference allele is whatever the mouse reference genome is, right? So for those guys over here, the reference allele, so this is a density. This is relative to, I guess the population mean. I'd have to look up what I usually did for the scaling of density, but it basically is relative frequency. So anyway, so we got the two warm colors, the orange and the red. Those are our two pools of soft selected lines. The blue is the stiff selected line. The purple is the ancestor. This is that is the allele frequency, the site frequency spectrum, the unfolded site frequency spectrum in the ancestor. And the black is Ayesha's simulations of the population. Basically this is what happened. These are the allele frequencies you would expect under drift after 90 generations and pool seek, right? So what you see is the simulation. So the simulation matches the ancestor very nicely. So if the only thing operating was random genetic drift, that thick black line, so the top and the bottom of the thick black line, those are the 95% confidence interval. So that's, we got a lot of data, right? So the simulations match the ancestral frequencies very nicely. And you also see the two soft selected lines look very similar to each other, right? There's a little bit of difference between the yellow and the red, but not very much. And you see there's a clear difference between the soft selected lines and the stiff selected lines. So the shape of the frequency distribution looks, the peakedness looks similar, but the soft selected lines are sort of pushed toward the extremes. So what that tells me is that the stiff, my interpretation as the soft selected lines are evolving faster, so they're losing, so the segregating alleles are being lost faster, which is pushing the allele frequencies towards 0 or 1. And the stiff selected lines are getting there, but they're going to get there slower. That is my best interpretation. And this is just basically a snip by snip, blow up of, one very little bit of that, just to show that, yes, for this, those are your two soft selected SNPs. Here's your stiff selected one. Here's the, so basically that is just that at a much larger scale. So, okay, so the conclusion, too much evolution to be explained by random genetic drift. Ergo, the population must have been under natural selection, which is what we would have concluded anyway, even without this. But, now let somebody try to argue. Okay, so, okay, this is a fishing trip. So we wanted to look at for causal variance. We said, okay, so how are we going to, again, everything is in complete LD, right? And we have no idea of haplotype structure. So what are we will basically pick the minimum change in allele frequency that we saw in the neutral simulations, absolute value. And so if the minimum frequency of the allele, of a change in allele in the real data set was greater than the most extreme change we saw in the simulations, that was a candidate selected locus. And then the minimum scaled allele frequencies, the scale, the frequency of the change relative to the initial frequency. And more than 10 reads had to have the minor allele in at least one sample. Or which is another, it's a terrible way of saying that. At least once the sample had to have 10 reads of the minor allele. And the frequency of the minor allele had to be less than a half in the ancestor. So that was our set of criteria for picking candidate loci. And we didn't come up with any. So that's what we got, right? We got, after all of these, all this filtering, we got one candidate variant. And one of the samples, it had a relatively high coverage, and the other sample, it had a low, minimal coverage. And I think it was a false positive. I don't know what it was, some weird thing that would have, I don't even remember what the gene was. It's a false positive. Okay. So take a brief pause. So now I especially, we were interested in, the what does the phenotypic plasticity we observe mean? And this is a figure structure I stole from George Yang. So O represents, so we got 3 environments, and what we're plotting is phenotypic value, size or something on it. And O represents the trait value in the original environment, which in this case is a stiff substrate. The plastic change is the difference. The P represents the plastic response of the trait in a different environment, before, at the beginning, right, of the ancestor. So this difference between here and here, that will be like growth rate, or spreading area on stiff, spreading area on soft of the ancestor. And then E here represents, this is the after evolution. This is the evolved environment, or the evolved trait in the, so this is what happens after 90 generations of selection on soft substrate. And the various arrows represent the various possibilities. I think I have, yeah, okay. So I mean, so if, so what we see here, so for example, and this is I think this is a way that a lot of evolutionary biologists, especially a lot of evolutionary ecologists, think about phenotypic plasticity, is this is giving the organism a head start. The environment, natural selection favors one phenotype when it's hot and another phenotype when it's cold. And if you have plasticity where it does one thing when it's hot and it's another thing when it's cold, that somehow, that change gives the, I say, we call it reinforcement. That's George Yang's term, but an evolutionary head start. So I think this is kind of what most of us would predict. If you have an organism or an entity that shows that kind of plastic change, if you now put it in the new environment and let it evolve, it'll evolve even further in the same direction. And I think that's what I would have predicted when I was a sophomore in college and first heard anything about evolution, right? The pink, that represents what I call perfect plasticity. Natural selection has got it totally dialed in. If it's hot, it's up there. If it's cold, it's down here. And now you let the environment, you let it evolve in cold, and there's no further change. That plasticity gives it, perfect. It's a perfect phenotype in both environments. Or you could see the blue, any of the blue, I'm not even going to bother with this. He says bothering with it. So it could be, now after you let the system evolve, it'll go back to where it was at the start. And or even further, maybe. We call that reversion. And if you see something like this, that could, this plasticity could even be maladaptive plasticity. So, okay, cell spreading area. How am I doing? I have no timepiece. Oh, there's a timepiece. Oh man, I'm going slower than even usual. All right.
Don't worry.
Okay, so I'm definitely not going to get to the seconds. Well, it's okay. All right. So cell spreading area returns to normal. So this, this is the plastic. Here's the ancestor in the original environment. Here's the ancestor when you give it soft substrate. after 90 generations of evolution, it's back to where it was. And thus, the stiff selected lines don't change. I think he got that backwards, but anyway, there's no difference. YAP, nuclear localization, and there's pictures of YAP. And if you're a cell biologist, which Tanmay is, you can't have a paper without these images of stuff being expressed in various places. But same thing, YAP expression returns to normal. Then this is the nuclear to cytoplasmic ratio of YAP. Cytoskeletal organization, you can take, I'll take Tanmay's word for it, that the cytoskeleton, which is the actin expression, returns to normal. So we're looking at this whole suite of traits in the soft selected lines, and they've returned to what the ancestor does on the stiff substrate. Okay. Cell spreading area. Okay, so phenotype. So I think I showed you a graph of this, but this is an image of spreading area. So the soft selected lines on soft spread a lot. The soft selected lines on stiff spread less. And so this is I think this is just, I'm showing you the same figure again. I don't know why that's like that, but okay, so another fishing trip. Did RNA-seq, and this is what this sort of biologist definitely wants to see the transcriptome. Okay, so we did a whole transcriptome RNA sequencing. Each of the 10 soft selected lines cultured on soft substrate, we grow the cells up on soft, we extract RNA, and we sequence it. Do the same thing for the stiff substrate. Now there's no pooling or anything. We're doing RNA-seq on each line, and we did 5 replicates of the ancestor. We'll see if there's any fish. Okay, so I saw about 7 of these plots and talking to everybody today. So you should be. So PC1 explains half of the variance, and that's so the squares. So blue and orange and blue. Those are the blue is ancestor on soft. 1 is soft. 308 is stiff. Orange is the ancestor on soft. Red is the soft evolved lines on soft, and green is the soft evolved lines on stiff. We didn't do the RNA-seq on the stiff selected lines. We only did the soft selected lines, but we did them on both substrates. And the circles are on soft substrate and the triangles are on stiff substrate. So PC1 clearly delineates the ancestor from all of the evolved line, from the soft selected lines. So clearly this is lab adaptation there's something about, doesn't matter if it's soft or stiff, they were grown on tissue culture plastic, now they're being cultured on acrylamide gels. There's something about the evolution, this is explains evolution on acrylamide. PC2, which explains, almost 30% of the variance. So there's 2 clear groups, right? And we call these the north group and the south group. But and it would, we were like, wouldn't it be great if substrate, all of the soft lines, or all lines grown on soft were up here, and all the lines grown on stiff were down here. Alas, that was not the case, as you can see, right? So we get clearly there's 2 groups, based on their transcriptional profiles, but it doesn't seem to have anything to do with, what substrate they're growing. Something else is going on, right? So
the other phenotypes you didn't see this that you showed before your whatever the 5 million.
So you mean we didn't do PCA, and we were just showing the means. Mm-hmm. But yes, we didn't. I didn't parse it down any farther than, yes, the means are very significantly different. I suppose I could ask, I didn't ask. So, yeah, was there significant among replicate variants? Is how I would phrase the question. There probably was, but it clearly was swamped out by the the difference in the means. All right, so yeah, okay. So we then asked if the genes that are differently expressed, do they, what do they do? So they've changed, they've changed significantly. We're only looking at the genes for which there's significant changes in transcription. Does the gene reinforce, right? So whatever the phenotypic does its expression get even more extreme than in the plastic, or than in the ancestor on the soft substrate, or does it revert? So basically, is the evolutionary response back toward the original, back toward the ancestor, or even more extreme? And so the first thing we say, averaged over all the lines, gene expression is significantly more likely to revert. So going back toward where you were, significantly more likely than going even more extreme. That's the first result. And genes in the north cluster are significantly more likely to revert than genes. Oh, whoops, that's the pointer. Okay, so genes up here were significantly more likely to revert than genes down here. Now this is, these are coarse features of the transcriptome. Okay, so this is even worse than a fishing trip. This is Elmer Fudd out to get Bugs Bunny, and we know that never works. Okay, so we look at growth and mechanotransduction pathways. These are sort of looking for the usual suspects, kind of. So we saw all of EGF growth, MPK, whatever that is, PI3K, mechanotransduction. So these are go categories, right? So all of those were downregulated in the north and went up in the south. And with one of them didn't change, and one of them was sort of kind of change. So what do we infer from all that? Okay, so what do we infer from all this? Okay, so first, the ancestor grows. Okay, so the ancestor grows fast on stiff and slow on soft. The ancestor exhibits a characteristic suite of phenotypic traits, plastic traits. Variable lines respond to selection, whereas clonal lines do not. So clearly, it is not just deterministic phenotypic plasticity. Soft selected lines grow fast. They have evolved, they've adapted to grow fast on soft, but they grow slow on stiff. So the pattern of plasticity has reversed. So, okay, so the response to select, so the way I interpret this is the response to selection is driven by initially rare genotypes that somehow do the wrong thing, right? By whatever mechanism the cell senses the stiffness of the substrate it's on, whatever signals it uses, those signals are telling the cell it's on a stiff substrate, and it should do what it does on stiff, and it should put YAP in the nucleus and make a cytoskeleton and all this stuff, right? And vice versa. And they also do this in at least two significantly different transcriptional ways. There's at least two ways transcriptome-wide speaking to skin this cat. Okay, so here's a picture that Tenmei and I struggled over for ages, and after I've gone away from it for a while, I always have to remember what the hell we're talking about. So this is the original, so we got the phenotypic value on the Y, we got our original environment, which is stiff, we got the plastic environment, which is soft, and then we have the evolved environment, which is soft. PC means plastic change. I had to look that up yesterday when I was. PC is plastic change. We explained this in the figure legend, but plastic. So the plastic change is now the direction arrow down there. And we've got some distribution of phenotypes, in our variable population. That's the blue frequency distribution. So if it was just plastic, if it was all plasticity, right? This is, we would just see everything kind of go back right to there, and that's not what we saw. because we know it's not plastic adaptation because our clonal cells did not do that. So there's a couple of possibilities. In the top here, we have, so this distribution represents the tail of that distribution. And now we have selected for these guys, and now what we see if we grow those guys up in the soft environment, the evolved cells in soft environment, we should sort of see that. And or we could be basically, this whole distribution could be moved down, and then after selection, we see that. Is that at least a little bit clear? He says anxiously. So this is what we saw, right? So these are the evolved cells now. So we're over here in E, right? So these are the evolved cells on soft. And what we saw is we didn't see this, right? So this would be, like if this distribution, if this distribution just moved up, we'd see, if you put them on, we'd see grow them on stiff, they would be even more, they'd spread even more, and they'd grow even faster. And that's not what we saw. What we saw here, so basically this is, we picked this tail that does the wrong thing. So for whatever reason, however the cells are whatever mechanism the cells are using to interpret what substrate they're on and what they should do, they get their signals crossed. And I'm sitting on soft substrate. I've selected for those cells who get it wrong. And it's 10 till, and I have been shut off. Oh, that's what, because, okay. So I think this would be, so it's 10 till 6. I think I will stop here. I will tell you a 30-second story. We did the same experiment in a different set of, so these are breast cancer cells. We saw exactly the same result. The variable lines responded to selection. The clonal lines did not. We saw, they did respond to selection. The trajectory of selection was remarkably similar. So again, we think we're selecting on rare cells. So what we did, we barcoded some of the cells, and we sort of followed the barcodes, and it's all very interesting, but it would take me another 20 minutes to explain it. What we saw that was different, we didn't see that pattern of reversal of plasticity. What we saw was the cells lost plasticity. They did the same thing no matter what. So in the first case, we selected for cells that are so dumb, we selected for cells that are dumb. They do the wrong thing. In the second experiment, we selected for cells that aren't paying attention. They do the same thing no matter what. Thought I put an environment. Anyway, I thank my friends and colleagues and funding agencies, and thank you all for inviting me, and I'm happy to take questions.